yields back. Thank you. And Mr. Dunn, you're recognized for 5 minutes and 8 seconds.
Thank you very much, Mr. Chairman. Thank the panelists for coming here today to inform us about your labs. I look forward to coming to visit you at the labs, and probably a lot more informative visit when you can actually teach me into place. I'm a physician. I'll tend to focus on healthcare in my questions. And if I could start with Dr. Kearns. Millions of people around the world have been helped with medical isotopes in the use of diagnosis, treatment, imaging, and other diseases, cancer and whatnot. We lack the capacity in the United States to produce all the needed isotopes, and some of our isotopes are in fact produced in highly enriched uranium reactors, which carries proliferation risks. Your lab is helping 2 companies develop new methods to make these isotopes in accelerators rather than reactors. I'd ask you to explain why it's important to the medical community to have these isotopes and to the patients to have these isotopes, but also why avoid the highly enriched uranium reactors.
Yeah, the last question first, fairly simple to understand. With the use of highly enriched uranium, the concern is one of proliferation and really safeguarding, if you will, the materials in a way that doesn't allow them to end up in the hands of those who might wish ill on individuals or nations, and so that's really the key component there. Certainly, you know, one of the primary examples, and the other laboratories involved should, I think, in the isotope program should also comment, but the one program that's been active at at Argonne has been the Moly-99 program, which is funded by the NNSA, working with a couple of industrial companies, if you will, to commercialize that technology. It's a great example, really, again, of industry laboratory partnerships and really driving towards a well-defined outcome, if you will. I think the why do that Moly-99 is pretty essential, really, in terms of how we conduct medicine today. It's critical for the United States because we don't have any domestic production of Moly-99 currently. We're really dependent upon foreign sources, and of course the reliability of those sources is from time to time of concern. And so
it's actually interrupted my practice on occasion.
Oh, has it?
And I'm sure I'm not the only one.
Yes.
So it's important for us to be able to get to those. Now, some of the isotopes simply cannot be produced in a reactor, in a accelerator. They need a reactor, and I know in recent years we've pushed to move from HEUs to low-enriched uranium reactors, and I understand that. I'm on board, but the low-enriched uranium reactors generate roughly 10 times the radioactive waste as the HEU reactors, and the United States has zero capacity to reprocess radioactive waste at this time. I believe I'm right on that. There's no MOX plants in the U.S. Dr. Peters?
There's not the capability to reprocess currently in the United States.
And the MOX plant that's been under construction in South Carolina is slow or off track?
It's behind schedule, over budget, and it's dedicated to reprocessing of the 34 metric tons of surplus plutonium.
Okay, so that's
so
all.
It's not going to reprocess HEU.
Okay, well that's worth knowing. Thank you. So I invite all of the panelists to bend your considerable talents and resources to solving the problem of reprocessing our domestic radioactive waste, which should not be treated as waste in the first place, but as a valuable precursor commodity. And I think we all know that the second generation radioactive waste that comes out of these reprocessed and reused, that you've changed the half-life of those that radioactive waste on the far end of the cycle to something that's logarithmically less, right? I mean, we move from hundreds of thousands of years, millions of years half-life to hundreds of years half-life. So I would encourage everybody to solve those problems. I know you can do it. I have faith in you. I'm going to sneak in on another question, if I can, on the ultra-fast, this is to Dr. Cow, the ultra-fast frame rate x-rays you use to capture molecular movies. Fascinating stuff. I've seen some of the work that's available. Can you explain how we use that information on a molecular level and how it might pertain to healthcare?
Okay. So what the ultra-fast x-ray does is you can take snapshot of a protein that typically these are targets for drug development, and in particular those on the surface of a membrane protein. They are very difficult to crystallize into crystals, so they tend to be very small. And so with this ultra-fast X-ray, you can take snapshot of these and you drop them down into the X-ray beam, you hit it, it disappears, but you capture the image. You do 1 million shots of these. You then reconstruct the three-dimensional atomic structure of that. And then you can use that to guide you to develop drugs.
Thank you very much, Mr. Chairman. I yield back.